like Josh introduced it, I have my background in microbiology and bacterial cell biology. But when I started my lab, I decided that I was going to work with these weird microorganisms that are also prokaryotes. They do not have a nucleus. That's all archaea. And when I give talks, I love to exemplify how wonderful they are, because most people never seen anyone doing archaeal cell biology. So today I'm going to talk a little bit how they're amazing mechano machines and how they respond to environment. We always talk about how microbes and organisms in general are great to identify specific molecules and signals and then respond to that, like phototaxi. Well, phototaxi for light, but chemotaxi is really well studied bacteria. But I will start with something that you know really well, but always strikes me to see the numbers. This pie chart, it's only related to eukaryotes. And then if you think like, how we approach science and like life sciences, basically have a complete underestimated number of 10 million eukaryotic organisms, probably way more. We know have isolated or sequenced or at least glanced at around 2 million of them, but including all the organisms taught today, flies, worms, and yeast, everything eukaryotic, only 50 organisms that we consider models. I'm not even going to bacteria in archaea. So that's one of the reasons why I switched to archaea, because I think that it's very important for us to apply all of our methods or approaches to understand nature more broadly. And I think that was a very good decision, and I hope you'll agree with me by the end of the talk. So what are archaea? Like I said, they're prokaryotes. They do not have a nucleus, but if you draw, a relatively recent tree of life, you're going to see the archaea way closer to eukaryotes than bacteria. So put in a very, misleading way, we actually are a branch of archaea, and archaea way more diverse in terms of phylogenomics and distribution and diversity in nature than eukaryotes are. And the reason why one of the reasons why I care so much about archaea is because they are so close to eukaryotes, is because we think that archaea basically originated eukaryotes, and one of the evidence for that recently, they just isolated, cultivated an archaeon that has 60% of their genome based on genes that they were thought to be exclusive to eukaryotes. They call them Asgard, and they have Loki, you have Thor, like all sort of superfamilies of heroes. But what is very interesting is that the prediction that archaea, or the ancestors of archaea, the common ancestor of archaea eukaryotes, would have these tentacles and its protrusions, and then that would increase the chance of engulfing a bacterium, confirm it when they saw that. So everybody is very excited to study these guys. And like I said, this is predicted to originate the first mitochondrion when an archaeon, something between Deepan and Uri archaeota, engulfed alpha proteobacterium and create a symbiont, and then the first primitive eukaryote emerged. The angle that my lab likes to study this is a mixture between cell biology and biophysics, a way to study how they are so flexible and how they can adapt to the mechanical world and engulf and contact things and interact. If I could put this, summarize my lab, basically we are always seeing how cells take on the insults we throw at them, usually mechanical ones, and then how the cells can organize the intracellular components to respond to that. So this is quite common in the tissue cell biology world, but this is quite not so common for the prokaryotic cell biology field that focus a lot on on chemotaxis and the biochemistry side. We are very interested in why they develop that and how they respond to mechanical side. And one interesting thing that is probably consequence of this weird choice of how to read out the mechano environment is that archaeal cells, even like in a very close relative of species, they have all sort of cell shapes. And then we use cell shapes as a proxy. So our lab grows over 200 different species of these guys. And they have all sort of everything. Some of them, they were sleeping in strain banks for over 20 years, and no one has ever put them under the microscope. So we developed methods and microfluidic approaches to actually image them and microculture them while we perform time lapses and advanced microscopy, super resolution microscopy to understand their cell biology. But then I see all these beautiful, like, shapes like the teardrop, like, I love it because it suggests us that there is some sort of like a symmetry going on to generate one daughter with like, with one curvature and another daughter has like a pole that is pointy. But cell shapes, all these cell shapes are not so uncommon, they're not rare. So why is Archaea so special? Maybe they are not. We got, for example, diatoms and algae, they have all sorts of these shapes as well. The whole difference between Archaea and 99.9% of all cells in all nature, and meaning animals are a classic exception of that. Most cells have very hard envelopes or cell walls. You go to plants, you go to bacteria, most of them, they do have an envelope that is hard and defined shape. And then I love that because these experiments that I do with the undergrads I teach to try to understand the physics behind cell shape and then how to manipulate that. So this is a vacuum chamber and a water, actually this is an air balloon. So if you take the air, basically the pressure inside the balloon is going to expand, and the balloon now finally you can make a square balloon. And then most cells use this kind of property to create any sort of shape. But animal cells and archaeal cells, they do not do this way. So archaeal cells, they do not have a cell wall. All they have are glycoproteins in the outer surface of the cells. So this is one example of a cryogenic structure with one of these polypeptide chains. And then the interesting thing is that they are filled with immunoglobulin subdomains. Some archaea have different numbers of immunoglobulin subdomains, but they are very similar to glycoproteins on the surface of viruses or some viruses. And then such capsids, they also out-associate no covalently to form a lattice, in this case, a P6 lattice, as you can see here. And this P6 lattice is going to sit flat on the outer surface of the cytoplasmic membrane and then self-associate no covalently to other P6 lattices and form a 2D crystalline lattice. I like to call this like or exoskeleton or protein origami of archaea. And this Z layer or surface layer is thought to be responsible for their cell shape, which is interesting because it makes them very plastic and they can change shape very fast, but they are also very mechanical, fragile. They can break anytime, and they do not have their siblings in the liver to protect them and then to create ECM. They are exposed to the environment as unicellular organisms. So one of the things that are very interesting because they're exposed to the environment, how do they face this environment? So one of our PhD students, Zach, from soft matter physics, he studied how one organism not only has different shapes, but also can interconvert from at least two different cell types. You can see here the rods, and they're swimming just like Dan show it. And then you can see these polygons are in the field we call discs cells. They do not have flagella. They have only cilia-like structures that we microbiologists call pili that adhere to the surfaces, and this is the pre-biofilm, substrate. These are the colonizers. So Zach wants to study how they interconvert between one and another. And you can see here this becoming rod. And it's very interesting that Zach says that during the transition that is very fast, is 1/10 of the cell cycle, they arrest cell division and they wait until they become completely rod to resume cell division cytokinesis, and they can follow up their life cycle. So one of the things that we were investigating in this project, and I'm going to talk much about it, is that the field thinks is basically carbon sensing. You have like a bunch of cells signaling, small molecules to each other, and they can decide which shape they can be. Other thing that we think a lot is about what Dan talked so well about stochastic noise. How you can have these different circuits can be being fired at any moment. But that's not what we proposed to study. Zach proposed to go and use microfluidics to see how the cells would undergo development when they have high shear and high current and high pressure in the fluidic, and then they have to react to that since they are so fragile. And then interesting is that even with conditioned media, with a lot of extracellular components, when we flow high and then we increase the shear, all of them respond by changing shape immediately. So this is a matter of under an hour. And then like Dan said, it takes some time more for them to start swimming. So different than what Dan said, they need a couple of generations. These guys don't want to create wealth for their future generations. They accumulate out for them, and then they stop the cell division. And then when they divide, they start swimming. So they are way more selfish than our grandparents were. The project, and then we are trying to understand what are the mechanical sensors that detect maybe you're wounding this S layer, and then there are factors that are going there, and Zach is working in a very nice way to build up this story. But this is not only the mechanoperturbation that we are studying. This is a project between a staff scientist, Teopi, and a PhD student, Olivia, that they are studying how confinement and compression of the cells, different than shear, help them and push into a developmental program that make them tissue. They are now multicellular. They have cell junctions and everything else that you would expect from any eukaryotic or animal or plant tissue. So everything started when we collaborated with Soana, the advisement, and together we built a microfluidic chamber that has 3 layers. So one of the layers we use to trap cells, and then the subsequent 2 layers we use to compress on-demand cells. So it's way simpler than it looks. Of course, it's way harder to build this than it looks, but to understand it's way simpler than it looks. So basically, you have cells being loaded, and by height, they're gonna be trapped by a soft polymer they call a PDMS. And they're just gently trapped. And then on the top valve chamber, we can apply pressure or not to compress cells with this PDMS. So when they're trapped, they look and grow and divide, looking just like bacteria here, for example. You can see the discs, and you can see some rods here. So they have a heterogeneity, but here they're okay. They're not suffering. They're growing as fast as in liquid bulk. However, when applied pressure on the top chamber and the PDMS deforms and squishes the cells, what we notice, and this is one cell, is that immediately they smush and they look like a pancake. And then I'm interrupt for a second. Please interrupt me anytime that you want. and sometimes people have like some questions about how to do the experiment. I don't need to finish completely everything that I have here in my slides. But the thing that we saw is one cell got smushed, and just like or a little bit similar to the shear stress where they stopped dividing, they grew isotropically, looked like a pancake, and they didn't divide it anymore. That was kind of like not so interesting to us. Maybe they're just stressed. But when this blob got way larger, we start seeing some carving out going on. This black, like, interfaces. And to finish, we saw that now that one big blob cellularized into multiple compartments. Each one is a cell, and the black borders are the junctions between them, holding them together. We were very surprised, and we didn't expect that. This is a labeling of the S layer of the proteinaceous outer layer, and you can see that they look like microcolonies, but actually connected to each other. So then I always like to show like how similar they look to plant leaf meristem. And because of that, we call it our outer cells. As you can see here, the outer cells look like larger, and I'm going to show that they're flatter. We call them apical cells, and the inner cells are called basal cells, based on the Merriam nomenclature. And as you can see, they look like a cap or like a street bump. And and if we photoactivate GFP only inside in the basal cells, they look more like scutoid-like. And it's very similar to scutoid cells. Oh, this is a lateral projection in STAB that is 40 nanometers projection. As you can see, before cellularization and after cellularization, you can see cells where they touch each other and where they do not. So you can see the difference between the height and volume of the cells. I'm not going to talk a lot about the structure and force distribution here, but basically they do have different volumes and they grow at different paces, but the ratio between surface and volume is constant, which is a hallmark of development for example, flies, where they also need to make scutoid cells to compensate this ratio.
Alex, if you stand for the fluid, like, does it part out like one per compartment, or is
that's a great question. Give me a couple more slides, and I'm going to show that actually this state over here, it's a coenocyte where they continue to replicate DNA, and then when they get to a certain ratio of volume per DNA molecule, they cellularize. Which is exactly what happens in some organisms. For example, chicken development, something.
Okay, what's the time scale like? So it's growing, and then what's it grow for, and then how fast is it?
So yeah, their generation time or doubling time is around 3 hours, and they take around between 12 and 18 hours to full development.
Okay. And so is the when it starts dividing, when the colony or the organism starts dividing, is it faster than normal cell division, or is it about the same time scale?
Same time scale, yeah. So what happens is that they stop for a while, and then they develop, and then they grow as a coenocyte, and then when they reach a certain size, they cellularize. But when they start growing at this stage, they are already growing at the same pace that in liquid. Yeah.
So in your system, can you measure or estimate the magnitude of the compressive forces? And like, could you put it in context for like where you might find forces in the same way?
Yeah, I didn't put in this presentation, but I can tell you that the pressure necessary to squish and develop them is the same difference. It's the same. So, and let me put this other way. When you have them in liquid as unicellular, they do have the same viscoelasticity than the mammalian cells in vitro. Very similar. And let's just put this in context that bacillus studies, for example, that then just talk about, has it's 1,000 times steeper than the cells. When they become multicellular, and then we use AFM for that, we could measure that the stiffness goes a little bit under epithelial tissue resistance. So the trend, like, when we study single cell, mammalian single cells and tissue, animal tissue is kind of similar. And that's what we think. In the environment, and I'm giving you the giveaway, in the environment, these guys live in for example, salt ponds that undergo salt crystallization all the time, and they can easily get trapped by salt plates and brine. So we think that because the outer cells grow so much faster and it's so larger, they can escape from that environment, and then the basal cells can sacrifice themselves. But we're still collecting brine from the environment to find if we can pick exactly this state of development. Anything else? Okay. So, of course, I talked about chicken. It's amazing how, turtle is also the same. It's amazing how there's not as close as, like, fish and mammalians. There's not enough, like, in embryo development studies on chicken and turtle, but basically they also cellularize like flies, in some terms, and they also make choanocytes. One more closer example to archaea, the holozoans, they are pre-animals. They're pretty close to choanoflagellates, for example. They do have, like, a unicellular and multicellular cycle where they make these choanocytes, they keep replicating the nuclei, and then when they get to a certain number, they explode and make a bunch of, unicellular, something similar to sporulation, but they're not making spores. So our question was, is this happening? Is this happening at this, exactly this stage that they're growing as like a one big blob and then prior to cellularization? So we tagged a component of the replisome, PCNA, and then when you see cells that are not compressed in the fluidics, you can see all these replication forks distributed in the cells as they grow and divide. And if we get the total fluorescence of PCNA on the spots and divide by the area of the cells and normalize that, what we get is something like that. So each line is a lineage. Is a progeny. It's not only one cell. We have like the mother, daughters, granddaughters, and so on. But the important thing is that the amount of replication across like a lineage never change. They always replicating the same amount per cell cycle. So with give and take. But when we squish them and force development to multicellularity, first, we see that they continue to replicate. And second thing that we see is that the foci become way brighter and way more numerous. And then when we quantify that, it seems that they ramp up twice as much replication and they stay put in a new steady state of replicating twice as fast DNA, which is still something we want to understand better. How come you need to replicate more if you have the same surface area per volume, ratio? So we do think that this is important when you think about eukaryogenesis, when you have small cells like bacteria or archaea becoming way larger, like yeast, and so on and so on. So diffusion starts to become more limiting. So maybe more replication is more necessary. We also see more protein synthesis that is not in the slice happening, it's upregulated. So it's an interesting adaptation. So it's a choanocyte, meaning it keeps replicating. Another thing that we are very interested in is that what triggers cellularization. What we did was something very simple that basically watch, measure, and correlate. So basically we see guys cellularizing and we use a balloon segmentation plugin that we customize it to tell when the symmetry of the balloon is broken by the junctions within the balloon, that is the colony. So when that happens, the computer knows and then start measuring the area of the balloon. So we have these curves that are growth curves. So the first point that you're going to see is the point in time on the x-axis that they start to develop. And then you have cluster area. And then if you get exponential fit, as Josh asked, the growth rate here is very similar to unicellular in liquid. But one thing that is like, you can see by eye, you don't even need fancier PC like analysis, PCA analysis. What we can see is that the time on the x-axis is very variable between 5 to 20 hours, like something like that, as Josh asked it, when do they develop after they squish it? But the area, it's not as nearly wide. So if you measure time and area from compression to development, you have time being 3 times more variable, 30% more than, three times more variable. The CV is 30%, while area is 10%. So that would suggest that they are not measuring time, they are measuring area or rotation in the envelope, or they need to get to a certain size before they trigger cellularization. But this is correlation. We wanted to actually test the hypothesis. So what they did was to put them in the fluidics and then genetically block division in one chamber while having control of the same strain in the other chamber. So these are parallel experiments. So you can see that as they grow non-compressed, the normal cells are dividing and they're multiplying, while the cells that inhibit, they do not divide, they look like blobs. And then when they get to a certain size that is closer to the size that we saw in the past graph, around 30 micron squares, Theope released the guillotine and compressed them. So the hypothesis here is, if it's time, it doesn't matter how big they are, they're gonna take time and then they're gonna just cellularize when they're way bigger. But if it's size, they should develop and cellularize immediately after compression. So, and that's what you saw. The bigger blobs, they cellularized right away after compression. While the normal guys, they took the time, as you saw before. So it's an interesting thing that cellularization doesn't need this adaptation to a higher replication time. There is no time for that. But probably increases fitness to give them time to adapt the whole physiology. And this is something that we want to test now. How more fit the normal cells or the tissue that is formed from the normal cells is compared to the guys that were developed when they were huge blobs. This is just formally quantifying what I just told you. And you see that the cell area cellularization doesn't change when they're already big compared to the normal ones, while time changes a lot. But one thing that we noticed, and then Dan was talking about when, it takes like nothing to make the basal body of the flagella. It takes 5 minutes. And then we know that when you express proteins, it can be very, very fast, but sometimes to assemble the whole machinery takes some time, like an hour or two. Something that we notice is that even in the big cells, so I'm gonna have to show you. Even in the huge cells, there is between 2 and 3 hours over here of a gap of most cells that they need to develop, which we think is a gene expression program changing for development. So we decided to do RNA-seq of the cells. So Theope went and she collected samples of each one of the stages of this development. Single cells, choanocytes, cellularization populations, and the mature tissues. And she sequenced the total RNA, mRNA of all of them. Oops, that's not, oh, here we go. So she collected and then compared all of them with the liquid culture, unicellular culture, without being compressed. And then in the beginning, when you make a choanocyte, you have a lot of things repressed, probably stress, make them stop growing. But as they develop, we see the blue points enriching more and more. And then now you have the multicellular genes being upregulated more and more. And then the volcano plot is good here, I think it's pretty clear, but I personally like a lot something like that to show that the difference between the downregulated genes and the upregulated genes. In the beginning, there's a lot of downregulated genes, and as they develop, they go away while they give space for other cluster of genes that are upregulated. And right now, that's what we are interested in, what is being upregulated to tissue. Okay. So we got all of this list of genes, like it's always 100 genes, which 80 were hypothetical. So we applied AlphaFold prediction to all of them and compared it to data banks pretty structure. And that was our surprise when we found a protein that was hypothetical HVO 2015 that has an actin fold. It was assuming that there were not actins in this archaean. So we were very happy with that. We even model filaments and the contacts, lateral and longitudinal contacts were conserved, which made us more confident. So we made a native GFP fusion. We tagged the endogenous gene under the native promoter. And then we named this volactin because of the organism, like that we're working with the organism called Helopherex volcani. So we call this actin volactin. So I don't need to tell you why actin is exciting for us, because actin can do a lot of things and remodel cells and remodel tissue. So because this is being upregulated in tissues, we wanted to see the function of this actin. So the first thing that we see that there's very little or no cables in the unicellular cells, non-compressed. But as the colony starts to develop and make a mature tissue, these cables pop up and then it becomes very, very visible, which is exactly what we see in the RNA-seq dataset. And then if you want to get a high resolution in 3D, this is kind of how they look like. In magenta, you see the out layer of the cells, while in green, you're going to see volactin labeled with GFP. They look a little bit like organoids, but I don't know. That's interesting. Then we start to see more about the dynamics of this actin. And we saw that the actin in the center of the colony, in the basal cells, they were very stable and fixed in one position, while the volactin cables or filaments at the outer surface, they were very, very dynamic, undergoing dynamic stability and treadmilling.
Do you see a deformation of the S-layer lattice or the actin starts to?
Yeah, so we are not sure right now. The data is not good, but we are very confident and doing better experiments. Actin is sensing tension, and we see cell-cell fusion and remodeling of tissue inside the tissue driven by actin. And I'll show you the phenotype of the knockout. You're going to see how interesting it is. But that suggests, this dynamic suggests that actually actin is doing something different in one cell type than the other cell type. So we also tracked the positioning. So this is a very young, early tissue. I cannot even call this tissue. This is right in the beginning of cellularization. And you can see that because the forces are still not well distributed, the actins are very parallel to each other. And this is well done in mammalian tissue, in actomyosin. But when the tissue becomes larger and larger, you degenerate this relationship between the cables. It seems that they now start to fill in the neighborhood cells and their positioning depending how much pressure they're feeling. Our hypothesis was that they become more and more important in the basal cells to stabilize them, since they are the ones that are presumably going to save the outer cells as they escape. They need to be stable. So we deleted volactin, and not a lot of phenotypes, interesting phenotypes in liquid or uncompressed. But when we developed them, the colonies, the tissues were unable to form basal cells. They start growing larger and larger and make the apical cells, they grow fast. But the center cells are called basal, they're the scutoid ones, they never form. Probably they form, but they collapse really fast, and we couldn't see that. But these guys started to die really fast as soon as they get to a certain size. So we truly think that this actin, so archaea don't have myosin, so there's no actomyosin to generate force. So we are still working to see what is the complex associated to actin cables to detect and sense tension now, and actually if it is doing that.
So Alex, where in the cell does the nucleation of the actin happen? Is it something that's happening at the membrane?
Yeah, they are selecting these guys. They are always, each end of the cable is connected to the inner leaf of the cytoplasmic membrane. So they're bridging the cells. Even in the unicellular cells, they're bridging. Looks like kind of stabilizing the cell, but we think that they are not bridging enough for that. That's why we have the hypothesis that they are actually sensing how much pressure is being applied to cells.
So there's the nucleator is probably the membrane.
Yeah.
It should be pretty cool.
Yeah, we added all the actin depolymerizing drugs and they work. We have the same phenotypes, but we don't have, we cannot detect just by using normal bioinformatics, the ARPs, ARP23, phalloidine. We cannot detect this stabilizing or nucleators.
Like spiral format probably more.
I don't think that, well, they need to have some sort of like stabilization or bundlers, because you see that the outer cells, they are very dynamic, and then so it seems like, well, in the eukaryotic actin field, this is well established. When you put actomyosin in fluidic chambers, purified in vitro, and you apply flow, they become more rigid because certain proteins bind more easily in the tensely stretched cables than the loose ones. So we think that something like that is happening. As they stretch and sense tension, factors are binding and then stabilizing it. And what is for the future? So we are doing a lot of things, and as you can imagine, now we can apply a lot of very interesting things that people in the embryology and tissue development and mechanobiology have been doing for decades. One of the things are some factors that we didn't detect by RNA-seq, but we caught by doing CRISPR-I. Our lab generated a whole genome CRISPR-I library. We also have a transposon library. So we detect some guys that do not change the RNA expression during the experiment. And we detected a protein that is also hypothetical, but it looks a lot like piezo channels. So we are working on this guy. When we deleted this guy, cells were a little bit sick, but when you look in the microscope, it seems that they are already developing even without compression. So they're blind to compression. They're taking up whatever is in liquid, and they think that they're in that situation. So now we can see this organoid-like, uglier organoid-like structures. And then we can study them in this, sense Another one that I'm very excited is that is very important in plants, but without any mechanism, it's already known that in mice and rats is important for brain development and sex determination, that is clasp. So clasps are beta propellers that bind microtubules and organize symmetry of some components in the cell during development. And the phenotype are very interesting. Instead of not making basal cells, they lose the organization of the tissue, like the scutoids become smooshier. And this is exactly the phenotype in plant meristem tissue. They lose the capacity of organized microtubules to determine where they're going to divide, and that make the cellulose cell wall more inconsistent. We don't know how this becomes here since we don't have a cell wall, and then we do not have microtubules. So it'll be interesting to see. Another thing that we are transitioning faster now is that what I call, so I told you that we have 200 plus species of archaea in the lab, and we have cell biology, genetics, omics, and now we're doing a lot of biophysics. So we are transitioning now in the lab what I call physics evo devo. That is basically getting the genome and doing omics in all of these guys following a certain phenotype, for example, tissue development, and then map and do comparative genomics to see presence and absence, and then see if we can understand how these traits evolved and where did they come from. So, for example, if you zoom in the genus that I talked about today, the Helopherex, in our lab we have around 12 different species of Helopherex. The one that I talked today is the third from the top, Volcani, but we already squished each one of them, and then we have species that do not develop, and that just die when we squish like minimally, and then species that do develop very similar to Volcani. And we are working on this species, and it's interesting that the genomes can be very similar. For example, these two guys here, they share 9 out of 10 genes in the genome. So we are excited to see which mutations happen maybe in promoter regions that change, that silent like the pathways that help for development of this species, and maybe we can reconstitute that just by one single mutation if we're left. Interesting. Oh, another thing that is very interesting, half of the pink ones that do not develop lost volactin. They do not have it anymore. So that's another thing that is interesting. How they survive, we don't know. So what is for the future, too? We're doing a lot of soft matter, material science approaches to understand the properties of the cell, but also the molecular and cell biology of this phenomenon. We're doing atomic force to understand, compare it to epithelial tissues, for example, how they distribute the forces. Traction force microscopy has a similar approach as Brilliant does. Brilliant microscopy, that is basically sending a stimuli and seeing how different parts of the tissue respond. We are also doing something that our lab is developed the expertise, that is single molecule tracking in live cells. So we want to see, when identify one of these components that we talk about, where do they go, where they should go, how long they stay, what is the kinetics and enzymatic reactions. So we can dive way deeper from the large, space and time scales, and go to the very, moment on this, judgments are made, to understand how we can maybe on demand create these organoids. And I think that that's all I had to do. I finished pretty fast. Usually I use more time than I should. But I want to thank the lab. It is a pleasure to see as a cell biologist, when you start your lab, you have 3.5 years ago, you have an idea of what you're going to do. and then you start working with such like, collection of different and weird people that they have their own desires and own ideas. And I really enjoy working with people that have different expertises and they bring their own contribution to the team. So today I see myself like, towards like, way more in evolution, cell biology, biophysics than simply trying to understand one process in the cell. But I want to talk as well. I want to thank because of my collaborators, because without collaborations and learning from other people, like I'm doing here with you, doing all that is impossible. We have to learn fast. Also, thank the foundations and that support financially our work. It's great to see that people are getting more and more excited about new model organisms and weird sciences and start learning where, how can we compare this like convergent evolutionary aspect of making tissues and how common it is to actually become multicellular, not only create a population that cross-talk and then can communicate with each other. And I would love to take any more questions if you have.